Let's shift now to the components of DNA, because they're important in how it operates. And let's start with the base. The base gives the identity to a specific point or position in DNA, and there are 6 shown here. There are 2 classified as purines, adenine and guanine, with a double ring structure, and 4 classified as pyrimidines, cytosine, uracil, thymine, and 5-methylcytosine, with single ring structures. The unmodified bases are often abbreviated by the first letter of their name, A, G, C, U, or T. Those that are found in DNA are adenine, guanine, cytosine, and thymine. And in RNA, we have adenine, guanine, cytosine, and uracil. Uracil is found only in RNA, and thymine only in DNA. And you can see that the difference between them is that thymine has a 5-methyl group, which distinguishes it from uracil. This is one way of distinguishing DNA from RNA, and we'll come back to that later because there are other ways as well. There's also 5-methylcytosine, which is a regulatory modification in DNA. See that the methyl group is very similar to the 5-methyl group of thymine? But the amino group at position 4 of 5-methylcytosine and the carbonyl group at the position 4 of thymine distinguishes these from each other. We started with the bases, but now what is the overall structure? The bases are attached to a pentose sugar. It's a 5-membered ring, and you'll notice that it's numbered 1', 2', 3', 4', 5', whereas the bases are numbered 1, 2, 3, 4, and so on. This distinguishes the numbering system used for the pentose sugar and the base. The sugar can have either a 2' hydroxyl, making it a ribose, or it can have a 2' hydrogen, making it deoxyribose. Ribose is found in ribonucleic acid, or RNA, and deoxyribose is found in deoxyribonucleic acid, or DNA. So this difference between RNA and DNA at this position is reflected in their names. The nucleic part of the name refers to the fact that these were found in the nucleus of the cell. Acid refers to the fact that they are attached in nucleotides, or nucleoside monophosphates, to phosphate, which is charged. So you start with the nucleoside, a base attached to a sugar, and add phosphates to the 5' carbon for the nucleoside monophosphate, nucleoside diphosphate, or nucleoside triphosphate. And we'll be talking about those through the course.